Hello everyone, my name is Thamer. In this short video, I will briefly highlight the key findings from our study on altered synaptic and astrocytic proteins and Lewy body disorders associated with GBA mutations. Parkinson's disease and Lewy body dementia are α-synucleinopathies, meaning they are linked to abnormal alpha-synuclein pathology in the brain. GBA mutations, particularly the N370S, are important genetic risk factors for Parkinson's disease and Lewy body dementia. Clinically, GBA mutation carriers may show earlier disease onset, faster progression, and rapid cognitive decline compared with non-carriers. The main hypothesis of this study was that PD and LPD cases carrying the GBA N370S mutation would show a distinct synaptic molecular signature compared with the wild-type PD and LPD cases. To investigate this, we examined post-mortem human brain tissue from 3 groups: 10 controls, 7 PD and LPD cases carrying the GBA N370S mutation, and 20 PD-LPD wild-type cases. Using immunoplotting, we analyzed 7 synaptic and astrocytic markers, 4 presynaptic proteins including SNAP25, VAM2, RAB3A, and synaptophysin, 2 postsynaptic proteins, neurogranin and PST95, and finally 1 astrocytic protein, VAMP3. We selected 4 cortical brain regions according to BRAC and BRAC Parkinson's disease pathology staging system. The anterior cingulate cortex is affected early, while the temporal and prefrontal cortices represent intermediate stages, and the parietal cortex is affected later. Our study highlights 4 main altered synaptic and astrocytic markers: SNAP25, VAM2, neurogranin, and VAM3. Among the presynaptic proteins, SNAP25 levels were significantly increased in the PD-LPD group carrying the GBA mutation compared with the wild-type PD-LPD group in the anterior cingulate, temporal, and prefrontal cortices. This opposing pattern suggests a distinct presynaptic signature associated with the GBA mutation. VAM2 was significantly decreased in both PD-LPD groups in the prefrontal cortex, suggesting shared presynaptic dysfunction in GBA-associated and wild-type disease. At the postsynaptic level, neurogranin was significantly reduced in the PD-LPD group carrying the GBA mutation, particularly in the prefrontal cortex. It was also reduced in both disease groups in the parietal cortex, supporting a role for postsynaptic dysfunction in both GBA-associated and wild-type PD-LPD. Lastly, the astrocytic marker VAM3 was significantly reduced in the PD-LPD wild-type group compared with both controls and the GBA-linked disease group in the anterior cingulate cortex. This finding suggests that astrocytic responses may differ between GBA-associated and wild-type PD-LPD within the tripartite synapse. Overall, these results suggest that GBA-linked PD-LPD is not simply the same disease with an added genetic risk factor. Instead, GBA N370S cases appear to show a distinct regional synaptic and astrocytic molecular profile. These key altered markers reflecting presynaptic, postsynaptic, and astrocytic changes. Finally, these molecular changes may help explain why GBA mutation carriers show earlier disease onset, faster progression, and rapid cognitive decline compared with non-carriers. and thank you for listening.